All right, cool. Welcome everyone to the fourth event this year. We're improving it step by step. Sometimes we have a bit of technical issues, but today it seems all working out fine. We used the right link for the conference invitation. Everything should be working. Just as a heads up, we are recording the session, so it's not just an online event. The point is really of recording the content here, specifically also of our guest speakers, the information. The aim of the NanoCon event series is really to spread awareness across the initiatives that we might have closely aligned to what Origami's Lab is aiming to achieve. So if you want to be visible on the recording, turn off the camera and ask questions maybe then after the session. For us, it's very important to do the recording so we can spread it out digitally and people can consume whenever they have time. I heard somebody making noises in the background, so please mute your microphone. One of the guidances we ask you to follow. Camera, if you want to turn it on, it's cool to see each other, but not a mandatory thing. You can ask questions even in between, use the chat function. We try to respond. And after the guest talk session, we will have a Q&A. We'll have a short interview session, and then we will be able to ask anything we would like related to Origami Lab or specifically the main focus is on the guest talk today. Ask our questions that we might have there. So quickly before we dive into the guest session, we will focus a little bit on Origami Lab for a couple of minutes, what it's all about, why this is taking place, and what's the main mission of the initiative. We're then going to quickly move to today's guest talk, presented by Gemma and Miguel. Both are co-founders of the Ersilia initiative, which is focusing on the democratization of AI for drug discovery, especially in a low-resource environment. Funding or also maybe also infrastructure-wise. Super interesting topic, really looking forward to it, and we're going to hear more afterwards. And then at the end, as mentioned, we have some time reserved for an interview, which will be done today by Christina from our sponsor, the University of Applied Sciences, FHNW. Somebody should shorten that name. Really difficult to pronounce every time for me. And we'll ask a few in-depth questions related to the initiative and the specific use cases. So let's move on. I would introduce real quick Origami Lab, just a few sentences, what it's all about and what the main mission is. There are three main, let's say, objectives or main areas. The first of all, first is it's a community-driven initiative. Fully focused on open source and open knowledge. It's really open for anyone to join. It's an ecosystem. It's an open platform where we host events like the NanoCon here, but mostly also focus on problems and open spaces in the medical research space and focus on technology. To achieve that goal, we partner with anyone who is really interested in and shares the view that science and research can be modernized and that there has been a leap of progress which could be utilized and would add significant advantages to all life and society overall. So we're looking for companies, scientists, entrepreneurs, organizations, startups, non-profit or open source initiatives. Whoever is really aligned with that view is really welcome to work with us or the other way around. We want to help launching or supporting initiatives and with really the goal in mind of helping each other of creating sustainable and long-lasting solutions and really focusing on cutting-edge technology that we have at our disposal this decade. A lot has changed since the last 10 years or 5 years. We believe that in this decade there will be significant progress done due to the combination of technologies that reached a certain level where it hasn't been there before. Speaking of machine learning or cloud computing, computational power, all these things, they are really, really on a high level these days, and are not being utilized to the full extent. So that's, for us, it's important to connect the pharmaceutical drug development sector or medical sector with those new emerging capabilities. So that's it so much for Origami Lab. A little bit on the open source and ecosystem topic. It's really about sharing knowledge across initiatives, across participants, sharing technologies, and really being an ecosystem driver where people find out about each other's work, initiatives get to know each other, and find touch points where they can collaborate or support each other, even if they maybe compete on certain aspects. It's a market economy where competition terms are relevant, and competing organizations find ways to work together on things that enable each other and compete where they really differentiate themselves, and where they can discover talented individuals in unique startups or open source initiatives like Miguel and Gemma have launched. So it's really about facilitating also knowledge, and for that reason we have established the NanoCon to promote everyone who's working in this domain. So feel free to introduce yourself if you want to work with us, and we're happy really to talk about how we can support each other or you specifically. So that's it about Origami Lab. On the NanoCon event side, it's our 4th event today. Here you can see an overview of previous events. We had actually four. Only one person from the ETH library is listed here. Excellent guest speakers and really topics. It was very diverse, very different application cases. And if you think there's a topic that you would like to present here with us, or you think someone is doing something really interesting, please shout out to us. We will reach out and see how we could get a participation organized. You can find all video recordings on YouTube. We record every session. We try to make clips. You can find the links on our website to each recording, so feel free to check it out. Always happy if people give feedback as well. So that's it for the Origami Lab introduction. We would then move over to our today's guest speakers. It's a very cool team. We have met Miguel and Gemma a while ago. Actually, Miguel has reached out to us over the contact form on the website, and we immediately connected together with the teams, had a cool initial discussion. Miguel and Gemma introduced to us what they're up to. It's fully in line with what we want to achieve with Origami Lab. They work on an open source initiative that should enable drug discovery in low income or low resource environment by using AI libraries or models which are either developed by themselves or co-developed with any other partner, or also just simply maybe provided by someone, and are utilized in drug discovery or drug development. Gemma, I really found it interesting, is in her private spare time also focusing on circus activities. So I still can't imagine what's going on there. It would be cool to see at some point, maybe a picture or video. And they're working together with Miguel on the technical side of things, but also are both coming from the domain in the pharmaceutical space, are experts in this area. And we're really happy to have Miguel and Gemma today with us, speaking about what they do at Ersilia, what particularly the problem is, how they think they are addressing it, and what exactly democratization is, and why the hell is this supercomputer in the church in Barcelona. So I will then head over to Gemma and Miguel, and yeah, thanks a lot for the talk, and see you later at the Q&A session.
Thank you, Victor. So
many thanks, Victor. Very nice introduction. Thank you.
So that's...
Yeah, so whenever so we'll do it this way, if you guys are fine with it. We'll present it as a tandem. So Gemma is gonna start and then I'm gonna continue. Do you see my screen?
Yes.
All good. All good? And you see my pointer, hopefully.
As well.
Yeah, all good. It's perfect.
Yeah, all right. Okay.
Okay, so it's up for me. Miquel, I think you chose the wrong presentation, though.
Oh, yes.
That's actually also, as we always point out, the aim of NanoCons to give those great initiatives the chance to practice also, speaking about what they do and taking it out to the public. Since there are so many brilliant initiatives doing a lot of great work, but sometimes miss one of the most important topics is to spread the word, right? So it's also
okay.
Stage for that.
There we go.
Yes.
Awesome.
Okay, perfect. So thank you everyone for being here. As Victor very nicely introduced us, we are the Ersilia Open Source Initiative. We are a small nonprofit. We started our journey in November 2020. And yeah, today we'll be presenting our work in the democratization of artificial intelligence models for drug discovery. Our focus is in low and middle income countries and infectious diseases. So the mission of Ersilia, why it was born, is our mission is to strengthen the research capacity against infectious and neglected diseases, because we believe that the world should have egalitarian access to healthcare, which is actually what is not the situation where we are at now. Ersilia was co-founded by the three of us. Eduardo is not here with us today, but he's the CEO of Ersilia. He's trained as a molecular biologist as myself, and he did his PhD in Cambridge in the field of metabolism, and now is also the chief scientific officer of Pocket, startup for drug discovery. And now Miquel did his PhD in Barcelona in the field of computational biology, though he's a chemist by training, and I'm sure he'll explain. And after some years of postdoctoral and research associate work in the field of computational biology and computational pharmacology, he decided to jump in on his own journey and start Ersilia. And finally, myself, I'm a molecular biologist by training as well. I did my PhD in the field of cancer and stem cells. And now I'm currently the project manager and also postdoctoral researcher at the Ersilia Open Source Initiative. And as Victor said, I have experience in the, let's say, social side of our small nonprofit, thanks to my experience in circus projects. So Ersilia started as a way to provide an answer to the prohibitive research costs for drug discovery. So as you all are very well aware, bringing a new drug into the market is between $1 and $2 billion. And that is very expensive for high-income countries, but it's simply prohibitive for low-resource settings. If we measure actually the research output in terms of scientific publications, in low and middle-income countries, they account for less than 10% of the total research output in the world. So they are really not able to produce research because research is very expensive. And this is not likely to change, because if we look at the number of institutions that are high level, meaning university institutions, in low and middle income countries, there is less than one institution per million people. So we are facing this huge drawback in terms of research capacity in these countries. And this has led to what we call the Western bias in biomedical research, which means that because research is being done in high-income countries mostly, it is also focused in the problems of high-income countries. So you may guess that high-income countries, the major burden of disease is due to non-communicable diseases, but infections are still causing around 50% of the loss of healthy years in low and middle income countries, whereas only 10% of the drugs, I think it's actually 7% of the drugs in development are targeting these disease areas. So there is a large number of people suffering from diseases that are actually not being investigated by the pharmaceutical industry. And it's not only about the number of drugs that are being produced, but it's also the type of research that is being done. So as the WHO already stated in 2013, and we fully agree with this statement, so is that all countries need to be producing research as well as consuming it, because the questions need to be answered locally to properly solve the problems of each situation of each country. So this is a big problem. We cannot solve it all, but the approach of the Ersilia open source initiative is to use artificial intelligence models, beginning with the drug discovery process, because basically artificial intelligence models can bring down the cost of drug discovery, screening a much larger number of molecules, and decreasing also the time to actually finishing a drug. So thanks to AI, we are able to produce much cheaper and much faster drugs that may be an answer for some diseases where the expected returns on investment are very low. We are very well aware, though, that artificial intelligence is cheap, but it has its own drawbacks. First of all, most of the artificial intelligence models that you can find out there are not open source, so they do not share code, so they are not reusable by many scientists. Many of them lack experimental validations, and the benchmarks are not clear, so this is also making it difficult to apply them in drug discovery pipelines. And finally, due to a lack of expertise, many AI models are seen as black boxes, and scientists need technical skills that are not available everywhere. And in many countries, actually, the training in computer science is very low, so they are not able to use all this wealth of information and resources that is already existing out there. A brief overview of the drug discovery pipeline, in case you're not familiar with it. Drug discovery, it takes about 10 years. There are many steps from when you identify a target for a disease to when you choose some chemical entities that may be active against this target, and finally optimizing those, moving on to the clinical trials and the approval phases of the pharma. So drug discovery can be helped by artificial intelligence at all levels, even to prioritize which genes are going to be targeted, to select which chemical leads can be used for a particular disease, or to optimize these leads so they acquire certain toxicity or pharmacokinetic properties. And even in the clinical trial and post-approval decisions, there is also models that can help us speed up this process. As examples of to show that artificial intelligence is already out there, is already being used, we wanted to share with you. I'm sure many of you have heard or have seen this AlphaGo documentary, which is about the first machine learning algorithm that was able to beat humans at AlphaGo, at the Go game, which is a very complicated game. So the same team that developed AlphaGo has now developed AlphaFold, which is an algorithm that folds proteins and has it's almost as efficient as doing the experiments itself. So this is really a huge advancement, as protein folding is one of the major drawbacks for drug discovery, to understand how the proteins are so that we can actually make drugs that target them. And this was done by the DeepMind company. And second, here you may not like the quality of it, but it's one of the first drawings that was completely made by a computer, just from what he learned from other human-made images. So the same way that computers can make images, computers can also design new drugs. In the field of molecular generation, one of the biggest news was last year when Exentia, which is a UK company, and this Japanese pharma claimed to have moved the first completely AI-designed drug to clinical trials for obsessive-compulsive disorder. And so the AI is already out there; it's being used in some companies. But what can the average academic researcher do? That's the question: how to translate all these great advancements that are happening out there to what all scientists can use? How do we bridge these gaps? So this is what we are trying now to do with the Ersilia open source initiative. We want to provide ready to use, meaning that a scientist without no knowledge on computing science can actually use these models in their day-to-day research. So basically, a typical question is, I have a certain molecule, I want to know what can it be used for. No? So I have my input is a drug, my output will be what is the target of this drug. And this can be answered by a simple or more complicated artificial intelligence model. Basically, what happens inside the model is that this model has some raw data, which must be processed, which takes huge of the effort. And then a model is trained based on this data, is tuned, is validated. If possible, it reaches a level of experimental validation. And finally, what we do, which is something that is lacking now, is we deploy this algorithm within the environment of the Ersilia model hub, so that a scientist doesn't need to see anything that is happening outside, but they are actually just able to give a drug and get the answer, which is a target. So following this rationale, we are creating what we call the Ersilia model hub, which will host 100 models by September, hopefully. That's our goal. Basically, there are models that define drug-drug interactions, models that predict the toxicity or the sensitivity of cell lines, or inhibitors of a specific targets, such as the Origami receptor. And to give you an example, we kind of divide the models that we have in the hub in 3 types of models. The first type is models that come from the literature. So what we do is we take the code of the models from papers that are published out there that are open source and that we feel are interesting to the scientific community and aligned with the Ersilia open source mission. And we take the code, we simply bundle it in our environment, and we deploy it. This example of the antibiotic, of a predictor of antibiotic activity, came out last year. It's super cool, but now what we have done is that you simply put your drug here, for example, halicin, which is what they found out with this algorithm, and it predicts with an interval of confidence whether this drug is going to have antibiotic activity or not. What we can also do is, instead of starting from the model, we take published data on interesting experiments. For example, here we present a huge open source effort to discover new drugs against malaria. In this paper, they screened 500,000 compounds, and they identified the new family with putative anti-malarial capacity. So we can take all these wealth of data that is sitting there and not being used, and we can train a model based on that data, so that now we have a new artificial intelligence model, where you can give the name of a drug, and it will tell you whether it's an active or inactive. And maybe even more interesting, what we also do is we partner with laboratories, mostly from low and middle income countries, and we train models based on their data. So we try to provide tailored models that help them answer their questions. So sounds very big, and it's actually maybe bigger than you think. Here we try to summarize the complex biological interactions that happen within the humans and actually all organisms. And this is the blueprint we are using to build up the Ersilia model hub. So we know that genes can interact with cells form tissues are related to diseases, which are also depending on the genes, and the genes are modulated by modulating pathways. So all these edges, all these interactions that we are picturing here is what we are trying to give an answer to with the specific models. Today, our focus is in compounds because that's our background expertise, and that's why we have started with a very specific focus on the first stages of drug discovery, but hopefully we can expand and cover all these edges that you can see here drawn. So to summarize what we do at the technical level, let me give an example of a simple artificial intelligence modeling that we do. So we have an experiment, which is an inhibition of Escherichia coli, a bacteria. We have molecules that are active and molecules that are inactive, as you can see in red and blue. Then these molecules, first step, they need to be summarized or translated into a language that computers can understand. So this is what we call a signature. So we featurize these compounds in ones and zeros, basically, so the computer can represent these compounds. And then once the computer has this information, we train a model that will predict that whether a molecule is active or inactive. And then once this model is trained, we can basically take a known molecule and predict whether the signature of this with the signature of this molecule, whether it's going to be an active or an inactive on an inactive compound. And luckily for us, what we do, how we do it is we use the chemical checker, which is a tool that Miquel developed during his postdoctoral research at IRB Barcelona, and that gives us a high efficiency in this process of featurizing the molecules for the computer to understand. So now I give the word to Miquel so that he can explain a bit more in detail the chemical checker.
Many thanks, Gemma. Thank you. So yes, I'm going to talk a little bit about the chemical checker, which is basically the backbone technology that we are using in our modeling pipelines at Ersilia. And the reason why I want to give like a bit of an overview of these technologies because as an initiative, we want to sort of prove that what we do is a state of the art and that has okay or very good performance in some cases. Okay, so basically the next following slides are going to be devoted to essentially convince you that we know what we are doing and that the kind of AI that we are doing is a state of the art. So the chemical checker is a technology, as Gemma has already introduced, that is devoted to representing small molecules in a format that the computer can understand. As you might imagine, there is a huge number of small molecules that we are dealing with, and with the chemical checker, we sort of try to organize the information that we have for them so that we can use it intelligently with machine learning. So I'm well aware that there's not many chemists here in the audience, but please don't run away. This is a small molecule, right? That we represent as an image, as a 2D image, and basically the shape and the type of atoms of this small molecule is what's going to determine whether it's a good molecule or a bad molecule for a particular disease. So how do we tell the computer that this molecule is this molecule and not another one? And how do we tell the computer that this molecule has this particular structure and properties? The way we typically do it, and the way that the whole field of chemoinformatics has been doing this for the last at least 30 years, is try to enumerate in this structure the kind of features that we identify. So for instance, we identify here a bond between a fluoride and a carbon. So we put a 1 because we identify this bond. We also identify an OH bound to a carbon, so we put a 1 that describes this bond, etc., etc., etc. So what we get with the computer is a representation of ones and zeros, as Gemma was saying, that is basically encoding or capturing the structural features of the compound. I hope everyone is with me here. So,
sorry, Miquel.
Yeah.
Can you hide this? Meet Google is sharing your screen because it's showing in the.
Can you still see my screen? Does it work now?
Yes, thank you.
Oh, good.
Okay.
Yes.
Very good. Sorry, I wasn't. This has been there for the whole presentation, I believe. I'm sorry about that.
No problem. No problem. I was just sitting here. It was itching me to say.
Okay.
But it's not a problem. It was.
All right. So basically, the idea is that we are representing this chemical structure as a numerical vector that the computer can understand, and most importantly, the machine learning models can use. Now, with the chemical checker, we are trying to bring this to the big data times or to the field of data science by basically acknowledging the fact that we not only know the structure of the molecules, but we also know much more about them. So, for instance, that compound that for sure you didn't recognize is a compound that is called alofrantrine that is used to treat malaria. This compound belongs to a particular family of molecules and has some particular activities, some of which are good and some of which are bad. Right? So capturing all of this information, which is available for many of the compounds, into numerical vector is what we do with the chemical checker. We try to go beyond just the field of cheminformatics, and we try to bring all the data that we have collected over the years as scientists into something that we can readily use in machine learning. So this was the work of 4 years, very painful years of my life, basically trying to catalog all the information that we can identify about roughly 1 million molecules that we can find in the public domain, and organizing this information into 5 levels of, let's say, increasing biological complexity, starting with the chemistry, but then going to the targets of the compounds, that is what proteins those compounds interact with, the network effects, that is the biological processes or the pathways that are affected in the cells, cell measurements, that is how the expression or the behavior or the morphology, the shape of the cells changes when they are exposed to these compounds, and finally, hopefully for some of them at least, some clinical information, right? So that is what are the indications or the therapeutic areas of the compounds, but also what are the side effects or the interaction with other compounds when we administer them to humans. This database structure is what we call the chemical checker, and something that we published last year in a good biomedical journal. So, as you can imagine, things are not so simple, are not so neat. There's a lot of work behind gathering all of this information from the public domain. I'm not going to bore you here. Long story short, now, thanks to the chemical checker, given a small molecule, we not only have the chemical information that I was describing before, but we also have a lot of information about the targets, the off-targets, clinical outcomes, cell-based assays, etc., about those molecules. Let me give you like a very, very, very tiny snapshot of this database. I think that here I'm putting less than 200 compounds. Remember, the database has about 1 million compounds, so this is a huge database of a very big size. Here in each row here represents a compound, and I've divided the rows in the 5 different columns that I was describing before. And what you can see here for each of the compounds is their signature, right? So their descriptor. So in the first compound here, you can see the description of the chemical properties of the compound, but also the description of the targets that interact with this compound, the response in cells, clinical response, etc. As you can see here, those signatures sort of can be organized in different therapeutic categories, and you can find some patterns already. You can see that the compounds that are treated in psychiatry, are used in psychiatry, for instance, they have some patterns that are characteristic, okay, etc. So until very recently, let's say until last year, the classical solution was only to use the chemical information of the compounds. Now, thanks to the chemical checker, we are using much more in our machine learning models, right? We are using the chemistry, but also all the rest. So we hope, with the hope that this is going to boost the performance of the eventual models that we build. Of course, it's virtually impossible to understand the database I was showing to you because it's all numbers. It's very abstract. One thing that we can do is basically project those signatures in the 2D space, in a 2D map, and see whether we can organize compounds in sort of groups or clusters. And indeed we can. Here I'm taking 10,000 compounds, drug molecules, and I'm organizing them according to the chemical checker signatures that we have, so that compounds that are, or points that are close by in this representation essentially have similar signatures, therefore similar properties. And you can see already some patterns. You can see this group here, which is very populated, is related to a particular family of compounds that targets a kind of receptor that is very important pharmacologically, which is called the estrogen receptor. These compounds, you can see it in red, they are indeed chemically similar, which is kind of expected in many cases. But sometimes we identify clusters of compounds here in blue or here in blue, where the groups that are assembled do not have similar chemical properties, but nonetheless they have a lot of shared biological properties. Here you can see a group of compounds that all of them target this particular kinase, and another group of compounds that all of them target this particular kinase, right? So, long story short, the chemical checker signatures allows us to sort of navigate the chemical space and identify regions that are biologically interesting. And we can do, at least in the eyes of a computational pharmacologist, we can do cool things with this kind of representation. So for instance, here, I'm taking 4 compounds as an example. As you can see, these compounds have very different chemical structures. They are apparently not related. Therefore, they are scattered here in this chemical structure representation, but nonetheless, they all have the same mechanism of action. They all kill the same cells, and they are all used in cancer treatment, right? So thanks to the chemical checker, we can identify that these compounds, they do have very similar effects in cells, although they have different chemical structures. Or the other way around, here you can see 2 families of compounds related to 2 very popular families of antibiotics. This one corresponds to this family of compounds, and this scaffold corresponds to this family of compounds. And as you can see, these compounds do indeed cluster together when we consider their chemical properties. They actually have the same targets, but when we administer them to human cells, they have very different effects, right? This might explain why some of these compounds are very safe and some of these compounds are not, right? Even if they apparently do the same things to the pathogen, they do not behave similarly in our bodies, right? So this, I hope, sounds very good and very promising, but we have a small problem when we deal with chemical data. And the small problem is actually a big one. Is the number of possible molecules that exist. Remember, we are trying to invent new molecules that will cure certain diseases. And when we are faced in front of the problem of inventing new molecules, the number of possibilities is essentially unattainable. Only considering molecules with, I think, up to 17 atoms, there are 10 to the 63 possibilities, right? So a chemist is basically overwhelmed with the number of possibilities and the number of design decisions that they need to take. Even if we are only considering the molecules that are today commercially available, I think we are over the 200 million, right? And for sure, we don't have data for the majority of them, right? Remember, the chemical checker is a very data-driven approach. We rely on public data, and if we don't have data, then the chemical checker is essentially useless. We only have binding data, for instance, which is the most essential kind of biological data, binding to certain proteins for 1% of the commercially available molecules, which is negligible. If we think of for how many molecules we have cell-based assays publicly available, this is maybe around 10,000 or at best 20,000. And if we consider drug molecules, which are the ones that have been made it to the clinics and for which we have clinical information, I think we are around the 5,000, right? So of the 200 million compounds that we can buy today, only for 5,000 we have, let's say, a lot of information up to the clinical levels. Not only this, most of the approved drugs, I would say roughly half of them, are inspired by natural products, which means that the chemical space is very, 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 very biased. It's very oriented towards certain regions, and the majority of it remains unexploited and unexplored. So a more honest view of the chemical checker would be this one, right, where actually we do have information, chemical information for many of the compounds, millions of them, but when we go to the targets level and all the way down to the clinics levels, we are missing more and more information, right? And this is a real problem, and for sure, it's something that at some point we had to tackle, right? If we want this tool to be useful for everyone, we need to circumvent this problem. And the answer to this was, as you can imagine, deep learning and artificial intelligence, and in particular, a very specific kind of neural networks called Siamese neural networks, that are known to deal well with scenarios where you lack a lot of data. Okay, the problem with Siamese neural networks, and the problem with this exercise in particular, in which we are trying to fill all of these missing gaps, is that it requires a lot of computational power. It requires a lot of computing time to train those models efficiently and with good eventual performance. So we had to train this over 1 week in a supercomputing center in Barcelona, and this is the one that Victor was referring to. It's a very nice supercomputing center, and the most distinctive feature of this computer, which I think that today is the second most powerful in Europe, is that it's inside the church, right? And the reason for this is that, as you may know, Barcelona is very hot, and inside the church, refrigeration is a bit easier, right? So we have this, I would say, is the most beautiful computer in the world here in Barcelona, and it's also very powerful. So we were very lucky, and we could use 1 week of this computer for training this Siamese neural network that I was mentioning. The consequence or the results of this neural network is that now we can have chemical checker signatures virtually for any molecule that we want. This includes, of course, drug molecules. As you can see here, I'm doing the same kind of projections that I was doing before, and you can really see how useful those signatures can be, right? So here I'm highlighting regions that correspond to infectious diseases, regions that correspond to neurology, cardiology, cancer, etc. And you can see that those signatures, chemical checker signatures that we have generated with this network, are very efficient at organizing the chemical space. This is the jargon that we use. Basically, are very, very efficient at grouping together molecules that have the same properties, or that will have the same properties in a certain specific in a specific therapeutic domain. But because we can do that for any molecule, we can also take the metabolites of different species, for instance, and see how we organize metabolites by species. You can see that human and mice, we do share a lot of metabolites, but those metabolites are really not very similar to Saccharomyces or wine metabolite, right? Okay, so we can do all these kind of things. Also with the tissues and fluids that we have in our bodies, you can see that the metabolites that we find in the placenta are different than the ones that we find in the liver or in the saliva, etc. Or even, and this was just for the fun, we can analyze food ingredients using the chemical checker and see that what the type of, as expected, the type of metabolites that we find in meat are very different than the metabolites that we find in fruits, for instance, or cereals, mushrooms, coffee, etc. Okay, so using the chemical checker is, so to say, a good way to get started with machine learning in drug discovery, because the representation of molecules that we have with the chemical checker is already very efficient. For example, 3 weeks ago, we teamed up with this initiative that is called Open Source Malaria, and basically the problem that they had, or the challenge that they had, was to invent new molecules that might be, they call them next generation treatment for malaria. Basically what they had already done, this group is based in Oxford, what they had already done is a couple of assays against the malaria parasite, which is called Clostridium falciparum, and they had identified some good candidates, but they were not satisfied with it. So they asked us if we could invent new molecules that would have improved properties against the malaria parasite, and so we did that. And we generated actually roughly 100,000 compounds. So here you can see a representation of 100,000 molecules that we invented with a machine learning intelligence. In blue, we identify regions that we predict are not going to be very active and therefore not very interesting. And in red, you can see regions of the chemical space that we predict are going to be very active and therefore interesting for them to pursue. Let me focus a little bit and even a little bit more. And you can see one of those molecules, right? Remember, we had invented 100,000 molecules. You can see one here. So we have a few chemists here. You will recognize that this molecule is not too weird and it's not too special. So maybe it's going to be easy to synthesize, and it's going to be easy to test in the laboratory if this molecule can have some successful activity against the malaria parasite. But I've chosen here this molecule random, but as you can see, we have many more. Okay? So, to summarize, we are a very young initiative. As Gemma has said, we started only a few months ago, but we already have some track record, especially when we are collaborating with applying machine learning and artificial intelligence to the problems of low and middle income countries and to infectious diseases. We have done quite a lot in this field that we call data-driven drug discovery, and this can be checked, some of it at least, in some recent publications from this year and last year. But we have also done some gene expression analysis. This is another field of computational biology, the analysis of gene expression data, to sort of understand the malaria immunology in children in Mozambique. We have also done some medical informatics analysis in Zambia, in this case, recently published in the Lancet Global Health. We're very satisfied with this. And we have also implemented our tools and our pipelines, at least in two institutions. One, IS Global here in Barcelona. They are devoted to basically organizing medical data collected from several countries, mostly in Africa, in a single and visually intuitive resource. We are helping them with this, and we are also doing some analysis, again in Zambia and Lusaka, to do automatic reporting systems from clinical samples that they are collecting. So that's the last slide. Just a few takeaways. We are a charity. We are called Ersilia, and we are tackling mostly infectious and neglected diseases of low and middle income countries. Our goal is to provide AI models for drug discovery via this model hub, Ersilia model hub, that we are building, and hopefully we will release right after the summer in September, containing 100 models. Ersilia is powered and backed up by the chemical checker. We are strong believers in open science, and we are looking for collaborations all the time. If you like what we do, you can also support us. And I think that's it. Thank you very much for your time and your attention. I hope you liked the presentation.
Awesome. Thank you very much, Gemma. Thank you very much, Miguel. It was, for me personally, it was super exciting. It is a very unique initiative I think you guys are doing. A lot of hands-on work, not just AI buzzwords, right? A lot of work has been done in such a short time. Thank you very much for the insights. I would then head over to Christina. We have not so much time left. I think we covered the most important parts. Christina, if you could focus maybe on one or two, three questions that would help Miguel and Gemma diving onto some topics that you think could be more interesting or have not been explained deep enough. So stage is yours, Christina. Thank you very much.
Yes, thank you. So thank you so much, the two of you. It was really very inspiring also for me. So what I was the whole time a little bit thinking about is you are doing this with the bioactivity signatures. And I was wondering, because for the bioactivity you also need the target. In what way do you futurize your target at the end? Because this is actually always the biggest problem. So this is what I'm interested.
Very good. So thanks. That's an important question. So how do you represent the target so that the computer can understand it, right? No final answer to this. We are trying two strategies depending on the kind of information that we have. One of the strategies is the simplest one, which is basically embedding or converting to a numerical vector the sequence information of the target. Okay, you take the sequence, you apply some sort of language model because a sequence is a set of characters, and you summarize this sequence as a numerical vector that captures the sequence properties of the protein. This is not enough in many cases, as you might know. So second strategy is to try to fold the sequence, right? Try to infer the protein structure and then featherize the structure, right? So try to then capture the geometrical properties of the protein once it's folded. Again, this is still an open problem, even if DeepMind have made a huge effort, huge progress, and so on. This is something that we cannot do always, and we cannot do reliably for all the proteins. But this is certainly a solution in many cases. We do that. On top of that, sometimes we try to focus on the regions of the protein that we think are relevant, and for this we apply some sort of structural analysis tools, like trying to identify which are the binding pockets that there are in the structure. And those are the ones that we want to focus the most, right? Because there is where the drug is most likely to be accommodated. So we do all of that, and this is our way to featherize proteins when the protein is new. When the protein is not new, for instance, human proteins, we know a lot about human proteins, right? So we apply essentially the same strategy that we are applying with the chemical checker, collecting all the functional information, all the cell-based information, all the pathway information that we can find for the protein into one single vector that is hopefully very informative, okay? But this can only be done, as of now, with proteins that are very well characterized.
Okay, so you have actually the same issue as everybody else.
Exactly.
Okay, so the pathways are actually a very good keyword, because I was interested, you were talking also about the pathways you probably have. Do you have incorporated them automatically, for example, the databases from Reactome and KEGG and so on?
Yes.
Or
yes, we do, and as you know, because I see that you've worked with these databases, the reliability of these pathways is variable, to say the least. So we need to assign some sort of confidence score to all the data that we are assembling, right? The good thing is that because we have so much data, we kind of can compare the data that we are gathering, the new data that we are gathering, with what we already know, and see whether it makes sense, at least statistically, right? So if we get a new pathway, we can compare it to the rest and see if it matches or not. And we do all these kind of things so that we can semi-automatically at least curate the data. But yes, we are collecting Reactome, we are collecting KEGG and others, as well as interactomes, that is large-scale protein-protein interaction networks from several resources. Yeah, I've been doing that for a long time. Yeah.
And what also interests me is, do you plan? For me, it sounds like you can then apply. You said like the AI or the machine learning model. Do you plan to just make it possible to apply one, or can we have maybe a workflow of different machine learning models in a sequence? Do you plan to incorporate something like that as well?
That's our dream. We are basically to go into the technicality here. Each machine learning model is a Docker container. That means that it can be installed or removed from your computer. And of course, if you want to run more than one Docker container at the same time, you can use what is called container orchestration, right? For instance, Kubernetes. So our dream is that to have a tool where you can say, I want to run this, and this, and that all together, and then you just click and all the predictions are going to be run simultaneously, right?
Okay. This is really amazing.
Yeah.
This is really amazing. I have many more questions.
Last question for you, Christina, because maybe we have
if
someone in the audience.
Maybe I can ask the audience if someone has a question from the audience until I prioritize my last question.
We have a question from Craig. Let me read it out loud. Hold on. I perhaps missed this, but I was interested to know more about the library of compounds you generate. For example, the malaria example. Are these compounds all novel? And how do you verify that these don't appear within the scope of patented applications of similar analogs, for example, within the Marcus structure? Then the goal of these hits, how you would aim to get them through clinical trials and to patients. Actually, I had a similar question in my mind.
Okay. Yeah, I can answer that if you want.
Of course.
So the algorithms that we are using to generate molecules are called reinforcement learning, and basically they have several constraints that the algorithm keeps learning and learning over them. And you can tell, for example, you cannot be similar. So in this case, I don't remember how many of those we put, but we can give them a list of molecules and tell them you cannot be equal to any of these lists. So if we are worried about patented drugs, in this case, it was not so much because we are really focused on a chemical space that is already well defined by the open source malaria project and is not being used by any pharmaceutical company, because actually, and this is related to the question, their goal is to find new drugs in however they can, and they had a good series that they then, it's all in GitHub, it's open source. They found out that this one of the hits they were working with was actually very similar to something a pharmaceutical company was working with. And so they discontinued that because they don't want to actually duplicate efforts. And basically, the idea of this open source malaria is to produce drugs without patents. So they do it. The guy that started this initiative, Matthew Todd, also has a private pharmaceutical company that has a new paradigm of, let's say, drug discovery. And what they aim to do is to simply produce the drug, and because drugs for infectious, for neglected diseases have already protection that is not patent-based, this would suffice for covering the cost of production. And so it's, let's say, it's a for-profit, but they don't aim to make any benefits of it, and because all the major steps of drug discovery are done in open source, they also don't cost so much money as they usually would. And you can read more about this new model of pharmaceutical company on their GitHub, or the company is called M4K Pharma, Miquel?
Yeah, M4IK Pharma.
M4IK Pharma, and they also have the same model as another pharmaceutical company that is finding new drugs for children's diseases. So it's basically patent-free and just recovering the cost of the clinical trials. Yeah.
Yeah, if I may add, one of that's actually a good idea. It would be nice to have a model that can predict the patentability of a new chemical structure and even its price, right? We haven't done it, but now that we get this question, I think it's an interesting model to include in our hub. Basically, takes as input a chemical structure and tells you whether it is protected or likely to be protected. This can be done, and also I think that because there is a huge database of chemical patents called SureChemble, the other thing that we can think of is trying to predict the price of producing this molecule when it goes into the market, which is a very complex problem, of course, but at least to get an idea, so that we can discard molecules that will be very expensive and therefore not feasible for lower middle income countries. Yeah, good ideas. Thank you.
Amazing. Thank you, Craig, for the question. We have one more from Oleg. Oleg, you could ask yourself as well, or shall I read it out loud?
Hi. Okay, I can ask it. I mean, you showed clustering, basically approach by chemical properties and biological properties. I suppose there could be some outliers that don't conform to general pattern, and how do you deal with those cases? And are there many such cases, or it generally quite separated groups of molecules.
Yeah, very, very important. So we of course, we do have a lot of outliers. Remember, we are gathering here hundreds of millions of data points. So we find outliers all the time. We are fine with discarding data. We actually discard, I would say, like 40% of the data that we gather just because we don't rely on it. Okay, so we are fine with it. Other teams or try to include as much data as possible. We prefer to have data that is of okay quality, and this means necessarily discarding a lot of outliers. Detecting outliers, as you might know, is a field of research on its own, right? So how do we define an outlier? It's already a scientific question, and it's not our main focus. So we are applying outlier detection algorithms to basically identify what molecules seem to be too rare to be included in this sort of systematic and general approach. Okay, and as I said, roughly 40% of the data points that go into the pipeline do not make it to the end because of this reason. Okay, this also means that we might be missing things that are very interesting, but it's a compromise. Yeah.
Mm-hmm. Yeah, so that's what I wanted to specify, because this is also some sort of limitation in case we have some new disease and we search in groups that we have, but maybe some efficient drug would be somewhere far, so we won't be able to find it with existing model.
Yeah, very good. So this is also related to this term in machine learning. It's called generalizability, right? Generalization power of the models, right? So you want to apply the models not to things that you have seen in the past, but things that will happen in the future, right? And for this, the models needs to be general, yeah? Not only memorize things, but also be able to predict. Again, it's a huge problem, and it involves a lot of parameter tuning and so on. We are doing our best to be able to predict activities against new pathogens, new diseases, new molecular families that have not been seen by the pipeline before, and therefore they are more interesting. Again, this involves a lot of work from the computational side, but can be tackled to some degree. The other thing that is interesting in this regard is this kind of new technologies in machine learning that are called few shot, one shot, or zero shot learning, right? You might have heard of it. Siamese networks which I have presented, are an example of this. Those are machine learning models that do not need a lot of data. Typically, they did in the past, right? But now there's this new family of machine learning models that are comfortable with a few data points, and they make good predictions even if you show just a few data points or even no data points, right? And those are nice technologies that we are trying to include. The only problem is that they take a lot of expertise to train, and also computational power. Okay, but yeah, this is something we are certainly doing.
Okay, and to this point of outliers, don't you have any maybe some systems that would get 5%, let's say, random values, random molecules, just in case if you hit on some efficient example? Would it be reasonable to include something like this?
Yeah, so this is exactly what we do. When we are training our models, we design what we call null molecules that we know they don't do anything, right? And we explore in those maps that I was showing, where do they fall? What you want is that they fall in a very particular region that is not contaminated by anything else. You want all the molecules that are basically sugar, right, that they don't do anything, into one particular region of the map, and the rest to organize and represent the molecules that are interesting. Okay, this is something that we monitor all the time.
Cool.
Thank you.
So we're way over time. I think it's time to wrap up. We will have Miguel and Gemma available on the Slack channel. I will share the Slack channel here, and you can also find it on the website, OrigamiLab.ch. You can join the Slack channel. We're going to be there chatting a little bit along, and I guess Christina will first bombard her questions on. Maybe one last question that I try to raise the topic a bit. Why specifically have you went as a biologist or chemist into this highly deep technical level? I mean, Miguel, you have quite a profound knowledge now of algorithms and statistics now, although you probably have not planned this in the beginning, I would assume. Was there a moment where you realized you need to get familiar with technology because it's a tool that accelerates certain things for you? Or why have you dedicated 4 years, which you described to be really tough to this task, right? And why you almost like became an AI expert, right? Implicitly.
Because I thought it was easier, Victor. I don't know.
You thought?
No, really. I
agree.
Really
yeah.
I really believe that that if we want to make molecules for diseases that are not going to give economic return, we need to make those molecules fast, and we need to make those molecules cheaper. Okay. Right? And the answer, I think that one of the answers that we have today is computational methods, right? Because compared to any experiment, they are much, much cheaper, and they can be applied virtually from anywhere and by anybody. Okay, so I think that this is I would say the main motivation, right? Okay. Moving a little bit away from experiments and making science that is cheaper and accessible to everyone. Okay, awesome. Thank you very much. We're at the end. It was a very insightful talk. We will share with everyone on the meetup page and also later in Slack the details of Miguel and Gemma. You will find the website link there and the contact details of both. Feel free to reach out to us if you want to get in touch with Gemma or Miguel, or directly if you struggle finding them. We're available here. Looking forward to the next event, potentially in 2 months now after the summer period. And wish everyone a great day. Hope you had a good time. You learned something new. I definitely did. Really inspired now for the rest of the week. And yeah, looking forward. Seeing you soon. Goodbye and all the best. Thank you.
Thank you, Victor.
Bye.
Bye-bye.
Bye-bye.
Thank you, guys.